I'm very delighted to introduce our speaker today. Dr. Stacy Andersen is an assistant professor of medicine at BU, sorry, I might butcher this, Chobanian and Avedisian School of Medicine, and the co-director of the New England Centenarian Study. She's a multiple PI of the resilience resistance of Alzheimer's disease and centenarians and their offspring, the RADCO project, and leads neurocognitive phenotyping protocols across multiple studies of longevity and healthy aging. I'm trying to read this very fast. As a behavioral neuroscientist, her research focuses on characterizing cognitive profiles of older adults, identifying factors associated with resilience to AD among centenarians and their family members, and developing novel methods of detecting cognitive change using digital technologies and behavioral features of cognitive test performance. That was a lot, and we're really delighted you're here, and the floor is yours.
Great, thank you so much. Really excited to be here today to tell you about several of the studies that we have going on, and then I'll tell you a bit about the way that we are characterizing cognitive function in our studies. So let's dive in. So the New England Centenarian Study was started by Dr. Thomas Perls back in 1995, so it's been going on for a long time. And during his fellowship training, he was taught the older you get, the sicker you get. So when he was assigned 2 patients over the age of 100, he expected them to have a lot of diseases and be confined to their rooms in the community that he was visiting. But much to his surprise, when he got there, they were 2 of his healthiest patients and hardest to track down because they were out giving piano concerts in the community, socializing with everyone. And so he wondered whether these were just 2 amazing 100-year-olds, 2 outliers, or whether there's something different about people who live to really old ages. So. And so that started the New England Centenarian Study, which, even though it's called the New England Centenarian Study, we've actually enrolled centenarians all over the United States and at times internationally. And to put in context what we're studying, at the start of the study, we were studying people who were born around 1900, and the median survival for that birth cohort was 67. 5% of the people born in 1900 lived to age 92, and only 0.2% of people born in 1900 lived to age 100. So we're really talking about living 30 or more years past the median survival of that birth cohort. So truly exceptional longevity. And over the years, we wanted to understand whether there was something different about these people who live to old ages. So we asked them to self-report the age of diagnosis of several different age-related chronic illnesses that you see listed here. And what we found was that centenarians on average didn't have a disease or had the onset of their first disease at age 95. So well beyond when their peers, most of their peers had actually passed away. And in comparison, the people who die at younger ages, they had a median age of onset of 71. Now, we don't just study centenarians. In some ways, they aren't even special enough. We also study super centenarians. These are people age 110 and over. And what you'll see here is that they had their first disease on average at 109. And so the centenarians really are delaying morbidity or the onset of a first disease. And so not only do they have a very long lifespan, but they also have a very long health span. But that also made us wonder whether they fit this compression of morbidity paradigm, which means that if lifespan is fixed at a certain age, but you are able to delay the onset of disease, then you must, by definition, be contracting the amount of time that someone's living with a disease. And when we looked at median percent of life with disease, our centenarians were only spending 9% of their lives with a disease, whereas the controls, it was about 18%. And then again, those super centenarians, it was only 5% of their lives. So they're not just adding years to their lives, they're adding healthy lives. And what we're trying to learn about is what contributes to healthy aging and that health span that almost matches your lifespan, regardless of what age you live to. And then we looked at their functional abilities as well, and we saw that 90% of centenarians were living disability-free into their 90s, living on their own. And in these survival curves, this is looking at activities of daily living, and being up near the top of the graph is better. And here again, you see that our centenarians are spending the vast majority of their lives with very good physical function. And the supercentenarians in teal display that the most. Then when you look at cognitive function, about half of our centenarians are cognitively healthy within 1 year of death. And this was quite surprising to a lot of people because everyone assumed that dementia was inevitable with a long enough lifespan, but that's clearly not the case. So this is performance on a cognitive screener. And again, you see that the centenarians are really living the vast majority of their lives with good cognitive health. And again, those supercentenarians in aqua display that the most. And then over the years, we've seen a lot of evidence that longevity runs in families. So our centenarians were telling us that they had a mother or father who also lived to 100, or an aunt and uncle. And we were noticing that their children were also very healthy. So we started studying the centenarian offspring. And over the years, we've seen that they have lower mortality than their peers. They have a lower prevalence of chronic age-related illnesses such as cardiovascular disease, hypertension, and diabetes. And they have a longer cognitive health span than their peers. So they are 46% less likely than their peers to be cognitively impaired at baseline. And we follow them over time, and we're seeing that they are 27% less likely to become impaired over 8 years of follow-up. So we can learn a lot from both of these generations. And so over the years, we became leaders in studying longevity, and we became one of the study sites of the Long Life Family Study. And the aim of this study is a little bit different. Instead of studying people who reach the 0.1% of survival, here we were trying to find families that had multiple people living into their 90s, suggesting that there is clustering for longevity within these families. And so there are 4 study sites, and we enrolled over 500 long-lived families with almost 5,000 people. And here is one of my favorite families, even though we're not supposed to have favorites, but they were featured in Time magazine, and 3 of the siblings in this picture are in their 90s, and several of them actually ended up becoming centenarians. And one of the sisters at the time stated, "None of us have canes." So pointing to how they even were noticing that they were healthier than their peers. And over the years, we've enrolled 3 generations in these families. So we have the siblings, we have all of their offspring, and then now we even have the grandchildren. So we actually are studying people from 18 to 110 plus. And one of the benefits of a family-based study is that we can really look at familial factors that contribute to healthy longevity. So not only genetics, although that really was the primary focus at the beginning, but also shared environment and shared behaviors that are passed down in families. And so over the years, we've visited these people all over the US and Canada 3 times in their homes. We travel all around the country. And we do things like carotid ultrasounds, ankle brachial index, a lung function test called spirometry, a bunch of physical function tests, gait speed, balance, chair stands, grip strength. We collect blood to look at genetics, and then circulating biomarkers, other health and body characteristics, and we do a neuropsychological assessment. And it's about 60 minutes long, so it is abbreviated, but it goes well beyond a cognitive screener. And we administer all the tests that you see here, and we administer them at each visit. And then a unique thing that we do is collect a digital layer of data collection. So we have this digital pen that you see here that looks and writes just like a ballpoint pen. But when we get back to the office, we can actually download the data from it and play back a movie of any test that the participant was asked to draw or write something. And then all of the tests, we use this fancy voice recorder, and we then get all of their spoken responses. And initially, I had started recording the neuropsych testing session really just to make sure that we could capture everything someone said. And now we're learning that there's a lot more that we can get from these recordings, which is really great. But We wanted to take everything that we learned in the Long Life Family Study, how to do these deep phenotyping assessments to truly understand and capture heterogeneity among the sample. We wanted to bring that to our centenarian studies. So in the past, our studies really just used self-report of medical histories and cognitive screeners that we did over the phone, and we wanted to go well beyond that. So with the two centenarian studies that we have funded right now, the Integrative Longevity Omics Project and the Longevity Consortium, we have now enrolled about 2,000 centenarians, their children, and controls. And we collect a wealth of self-reported questionnaire data that you see here. We're doing fasting blood samples and stool samples, and we're generating a wealth of omics data. And we started the study right before COVID, and we had planned to travel all over the US. We actually did for the first few participants, but we quickly had to shift gears because, of course, centenarians were at the most risk of being impacted by COVID. And so we developed a remote assessment where we send out a box of equipment so that we can test someone who lives anywhere in the United States. And it includes a touchscreen tablet like you see here. And we have configured this tablet so that our staff can remotely control every aspect of it, from connecting to Zoom to increasing or lowering the volume for the participant. The only thing they have to do is press the power button. And then we include some other equipment in the box so that we can walk them through how to test their blood pressure and their temperature, how to use this dynamometer so that we can get a measure of their grip strength. And then we also send them an activity tracker to wear for a week, which collects hundreds of variables. And then we do about 90 minutes of cognitive testing, all of the same tests that we administer in the Long Life Family Study, plus a few extra that I've highlighted in purple. And then again, we have that digital layer of data collection. Even though we aren't seeing people face to face, we can still send them this digital pen and send them the voice recorder and get all of the same data. And then with all of these data, it really allows us to, as I said earlier, capture heterogeneity in our sample. So one of our more recent projects is focusing on cognitive superagers, and I'll tell you a little bit about what those are. So probably the most well-known study of cognitive superagers is led by Emily Rogalski in Chicago. And we all know that on average, there is a decline in episodic memory performance with increasing age. And staying within this gray shaded region is considered successful aging. You're within what's expected for your age. But what you'll notice is that there are these people who always are performing above average, even at age 80. And this really resonated with me because we know that our centenarians, some of our centenarians and some of our offspring are really high performers as well. And so these are people who we call cognitive superagers. They are performing at the level of someone 30 years younger. So these two 80-year-olds that are shown here are actually performing even at the top level of 50-year-olds. And the reason that there's a lot of growing interest in cognitive superagers is that they don't just have better cognitive function, they have better brain health. So in this image that you see here, great, the areas in red and yellow are areas that are of the brain that are thinner in 80-year-olds, average 80-year-olds, when you compare them to average 50-year-olds. But in the bottom panel, what we see here is when you compare these 80-year-old superagers to average 50-year-olds, there are no thinner areas. And actually, there's a tiny little area in blue that is thicker in the 80-year-olds in the anterior cingulate cortex. So there's something different about the way their brains are aging. And other work has shown that they have stronger functional connectivity in the default mode network. They have half the annual rate of change in cortical atrophy. So even over time, they are different. And then this other panel shows some specific types of neurons that are more plentiful in the superagers. And so we're really trying to learn what contributes to sustaining very high episodic memory across the lifespan. And as I mentioned, we know that centenarians, some of them at least significantly delay dementia onset, and their centenarians are also showing that same pattern. And so we expect that they are enriched for cognitive superagers. And so that's what started the RADCO study, which stands for Resilience and Resistance Against Alzheimer's Disease in Centenarians and Offspring. We want to find these high performers and understand what's different about them compared to average performers. And so we dive even deeper into their data. We're looking at blood biomarkers. We're trying to get them to go in for high-tech neuroimaging, and we ask them to participate in a brain donation program after they die. And then with our collaborators at MGH, we're looking at the underlying biological pathways that contribute to that. So looking at genomics and transcriptomics. And then we have induced pluripotent stem cells from these people. So we actually can grow out neuronal stem cell types or neuronal types, cell types in a dish, throw amyloid at them and see how they react differently. Do they clear it differently? Do they have different inflammation patterns? And the unique thing that we can actually differentiate with centenarians is that we can differentiate between resilience and resistance to Alzheimer's disease. Most studies focus on resilience to Alzheimer's disease. So we know that advanced age confers a high risk of AD pathologic change, which in turn confers a high risk of cognitive impairment. But we also know that there are a lot of people who have AD pathology in their brains, but still maintain good cognitive function. So it doesn't affect them the same way. And we say that they are resilient to Alzheimer's disease. They're coping better with that pathology in their brains. But we have some centenarians who don't even have the buildup of amyloid and tau in their brains, and we call them resistant to AD. So for some reason, they're just able to block that buildup, even though advanced age is the #1 risk factor for Alzheimer's disease. And you really can't study that in 80-year-olds because it's possible that they just haven't lived long enough and they will go on to get Alzheimer's disease. It's hard to say that a centenarian has not lived long enough. So now let me talk a little bit about how we are characterizing cognitive function in our studies. So kind of at the most global level, we have clinical case conferences in the Long Life Family Study where we bring together neurologists and neuropsychologists and geriatricians, and we review all of the testing data that we have. We review how they performed on a brief neuromotor exam, and we review data that we have collected from their informants, someone who knows them well and can tell us whether they are having difficulty in their daily life. And then we assign a cognitive status of normal cognition, mild cognitive impairment, and we tried to identify the types of mild cognitive impairment: amnestic, a more memory profile, or dysexecutive, a more executive function profile. Whether or not they meet the criteria for dementia and what we think is causing their dementia. And then we rate the severity using the clinical dementia rating scale. So these are kind of a way to bring together all of the data that we collect from people in a way that's usable for many of the groups who are doing analysis of our data. But of course, I'm a behavioral neuroscientist, so I feel like it misses all the wealth of information that we get from neuropsych testing. And so one thing that we've done is look at signatures of cognitive function, so how people perform across the different tests that we administered. And this is a heat map here where the boxes in blue show performance that's lower on average, and red is doing much better than the average of the sample. And you'll notice, and this is hierarchical clustering, so this is groupings of participants who all share the same neuropsych profile. So this group 4 here, they are performing poorly on all of the tests. And group 11 is performing above average on all of the tests. And then you have these interesting profiles in the middle. Some of them are more reflective of an Alzheimer's disease type syndrome. Some might be due to other causes. And what we can do with these profiles is actually annotate them with the wealth of other data that we collect in the Long Life Family Study. So now we can see which profiles are related to test performance at visit 2, or risk factors and blood biomarkers that we collect, or polygenic risk scores. And so this cluster 11 here, who did very well on all of the tests, they actually have higher educational attainment and they are enriched with genes that are related to cognitive performance. What you'll notice here is this group up here has a high percentage of smokers. And when you look down to their profile, they have average to slightly above average memory performance, but they're doing more poorly on the working memory and executive function tasks. And that fits with what we know about the effects of smoking on cerebrovascular disease. And so here we can learn a lot more about what's contributing to these different neuropsychological profiles. But all of those use the traditional scoring. And we know that brain changes begin up to a decade before we can classify people as having cognitive impairment or dementia using those traditional scores. But there's a lot of change between a healthy brain and one with severe Alzheimer's disease. And from this very well-known hypothesized figure of the changes that are going on within the brain, we know, or we believe, that amyloid and synaptic dysfunction and tau changes are happening much earlier than where we can measure cognitive function changes. But what if we could change where we can actually detect cognitive function changes? Then we could potentially intervene much earlier and understand what's causing the more downstream changes. But how do we move toward earlier detection using cognitive testing? So I was trained partially by Edith Kaplan, who's considered the godmother of neuropsychology. And she developed the Boston process approach, which is this idea that if you don't look beyond just traditional right or wrong scoring, you miss a wealth of clinically meaningful information. So looking at how someone goes about completing a task, what strategies do they use? What errors do they make? Those are the things that are likely to show up as different well before someone starts scoring poorly on a test. And this is what neuropsychologists do. They're really attuned to picking up these behavioral differences among people who maybe are at those most early stages of cognitive impairment. But it takes a lot of training to detect those, and it's very time-consuming and cumbersome. So here's where we can integrate digital technologies, where we can get a level of precision that potentially can even go beyond what a human being could detect as different among cognitively healthy people. So back to our digital pen. Which I mentioned earlier, allows us to actually play back a movie of how someone performs a task, like asking them to draw a clock, put in all the numbers, and set the hands to 10 after 11. But it also gives us a data file of time-stamped X and Y coordinates. And from that file, we can start to parse out different parts of someone's performance. So we can actually separate out the amount of time that they are putting ink on the paper, the amount of time that they are drawing. That's more likely to be related to motor speed. And then we can actually look at the time that they are holding the pen, not drawing, which is what we believe is when they are thinking. So when someone draws the outer circle of a clock, they usually pause for a second, then start putting in the numbers on the clock after they decide how they want to best go about that. And that is what we call thinking time. And then you can even look at how someone's speed changes over the course of a test, or looking at very specific components of how they complete the test. So coming back to our clock drawing example, there are very obvious differences when you're looking at clinical samples. People with Alzheimer's have very different clocks than people with Parkinson's disease, and those are very different than someone who is cognitively healthy. And I would say you probably don't need digital technologies here because it's very obvious. But what if you have two cognitively healthy people? What is different about these clocks, or what is different about how they went about completing the clock, that might differentiate someone who's going to go on to have cognitive impairment versus someone who isn't? And so I did a study with a doctoral student, Meng-Shin Du, and we looked at 1,500 offspring from the Long Life Family Study and 500 of their spouses, who were on average 69 years old. And we looked at some of these very subtle features of the clock drawing test. So we looked at clock diameter, the number of pen strokes that they used, whether they used anchors, so the 12, 6, 3, and 9, which a lot of people do to help them space the rest of the numbers correctly. We looked at that time after they draw the outer circle before they put in the first number. That's called the post-clock face latency. We looked at how long they paused before putting in their first hand as they're thinking about how do you represent the time 10 after 11 on this clock, the time that they paused before putting in the second hand, and then looking at that think time variable that I mentioned. And we saw many significant associations with age, sex, and education, just like you would with traditional scoring of a cognitive test. So higher age, male sex, and less education was related to having longer latencies. So thinking longer about how to do those components of the clock, and was associated with not using anchors. But here's where it gets interesting. So then we wanted to look at genetic risk. So these are cognitively healthy people. Some of them have an ApoE2 allele, which is related to a lower risk of Alzheimer's disease. In those people, they had lower total time to complete the clock, but they also had a lower sum of those pauses. So they didn't need to pause as long to figure out how to correctly do the clock. People who have the E4 allele, which is the risk allele for Alzheimer's disease, they had a smaller clock, but perhaps even more importantly, they had longer thinking time. So they were pausing more. And then we wanted to look at this factor of familial longevity. So we compared the offspring in those families to their spouses. We use the spouses as a control group because they share a lot of the same environment and behaviors as the offspring, but they don't have the genetics. And what we found here was that the family members of these long-lived families have a shorter post-clock face latency. So a shorter pause after drawing that outer circle. And so what this is telling us here is that these digital metrics are very subtle, early, potentially early markers of maybe potentially cognitive impairment. We need to follow them over time to see if that's actually what happens. But there are subtle differences related to genetics and familial longevity that it's picking up on that you would not see in traditional scoring of the clocks. So another example of work that we've done is looking at digital pen data from the digit symbol substitution test. And this is a coding test. There are many different versions out there where participants are given this grid and they're asked to fill in the empty squares at the bottom using the code at the top, and to do it as fast as they can for 90 seconds. And we know that as people start to learn the code, they get a little faster. Sometimes they get tired from doing it for 90 seconds or they get bored with it. And so I wanted to look at how their time, writing time and thinking time on this task changed over the 90 seconds. So not over a very long time. Wasn't even sure that we would pick up on differences. So we looked at every 8 boxes of symbols and chose the median time from those. And this was done in almost 1,600 long life family study participants with an average age of 70. And we did hierarchical clustering here again, and we found that there were 4 groups of performance when we were looking just at the thinking time. So how much time they were holding the pen and not writing. And it came up with 4 clusters. So one was a cluster that got slower over time, potentially due to fatigue or getting bored with the task. There were 2 clusters that were pretty stable and contained the majority of participants. And then there was a cluster that got faster over the course of the test. And actually the slowing group and group 4, the quickening group, were the ones that actually had poor episodic memory. And so you would kind of think that the quickening group would actually be a good thing. They're learning the key to the code over the course of the test, but actually they started out the slowest out of all 4 groups. So they really had difficulty engaging in the test at the very beginning. And so it was easier for them to get quicker over time. But now when we look at writing time, we again come up with 4 clusters. Interestingly, it's not the same people. So it was actually very unlikely that someone who was slowing in thinking time was also slowing in writing time. It tended to be different people. And what's unique about these clusters is that the slowing group, it was associated with poorer physical function. So those people had poorer grip strength, slower gait speed, and more perceived physical fatigability. But it did not, these clusters were not related to episodic memory performance. And that really tells us that by separating out the thinking time and the writing time, we're able to get at cognitive speed versus motor speed in one test. And then we looked at whether these clusters related to change in cognitive performance over time. I think it was a relatively short amount of time, only three years, yes. And the ones that are starred here are the groups that had more cognitive change over time. So saying that these clusters may have actual meaning for predicting who's going to decline over time. And then, as I mentioned, we collect digital voice samples, and there's a lot of work showing that lexical features and acoustic features and temporal features of how someone speaks are related to cognitive impairment. And I am very curious in how we can pull some of these out from cognitive testing and add to the rich data that we're already collecting. And one of the great things is that there are a lot of open source tools that are already out there to extract digital metrics from these types of files. So we're trying to figure out ways to use things that already exist to add more data to our rich data sets. And not surprisingly, one of the tests that I thought would be greatest for this is the logical memory test, where we read a story to someone. It's a very short story, just a few lines. Then we ask them right away to say it back to us. Then we do other tests for about 30 minutes, and we ask them to recall the story again. And what you see here is the traditional scoring. And there's a whole grid of what words or word substitutions someone can get credit for these different pieces of the story. But what you see on the bottom part is someone's actual recounting of the story. And in red, it shows what they got points for, but there's a whole bunch of text in black that they don't get any credit for, and it's really not considered in the traditional scoring. And so it feels like there's probably, or could be, a lot of meaningful information there. And so we have some studies going on that are aimed to identify subtle linguistic features from these responses that are associated with cognitive impairment. And I have a doctoral student, Seho Park, who some of you may have heard his presentations. So I'll just quickly summarize some of the work that he's been doing. But he used one of these open source tools, linguistic inquiry word count, actually was developed more for extracting emotional valence from text. And we're using it here because it was available and could tag some of the responses, and we could get metrics from them. So we have over 700 voice recordings from Long Life Family Study participants. Only about 16% of them have cognitive impairment because this is a healthy aging group. So we transcribed those recordings, and we did feature extraction with LIWC. And it quantifies grammatical features, things like words per sentence and pronouns, but it also quantifies the use of words in certain categories. So you'll see here, it has a category called work, and some of the examples of words that it would tag as a work word are job, majors, and Xerox. And some of these word categories overlap with things that we talk about in the Anna Thompson story. She was employed. So it actually, in that way, worked out pretty well for us. But we ended up using 51 of the variables that they have available. And here you can see the list of features that were associated with cognitive impairment in comparison to people without cognitive impairment. And in both recall conditions, immediate recall and that 30-minute later delayed recall, these 3 features, negations, words related to biological processes, and work, were both associated with were all associated with cognitive impairment. Then you have some that were only associated in the immediate recall condition and some that were only associated in the delayed recall condition. And what this tells us here is that we might actually be able to delve a little bit deeper into what strategies people are using in immediate recall to remember things and how those change over time for delayed recall. I don't have much more to say about it at the moment for there, but it's very interesting that those are not exactly the same. Yes. So then there were many features and they all had small effects. So we borrowed an idea from genetics of constructing a polyfeature score, or in genetics, it's a polygenic risk scores. And here we wanted to combine the features that were nominally associated with cognitive impairment into a score. So we have an immediate lexical polyfeature score and a delayed lexical polyfeature score. It's a mouthful. And you can see there's quite a range in this histogram here from a score of -49 to a max of 85, with the highest values being most associated with cognitive impairment. And each of these scores was predictive of cognitive impairment in the sample, so they can be used to distinguish between people who are cognitively healthy and those who are impaired. And here you see that they are not exactly the same thing. The correlation was only 0.62, so they are moderately correlated, so they have a little bit of overlap, but they are actually individually meaningful. And we looked at how well they were able to classify cognitive status. And there are a lot of precision recall curves that you see here, which we used because our sample was highly unbalanced. We had a lot more people who were cognitively healthy. And the immediate PFS score and the delayed PFS score were pretty good at distinguishing between cognitively healthy and impaired, but it really wasn't that different than using a traditional logical memory delayed score. And actually the traditional score was better at 0.84. But it is pretty similar to using a cognitive screener in demographics at 0.80. And then actually the best model included traditional scoring and the delayed PFS score and demographics, suggesting that this PFS score is adding something beyond traditional scoring. Maybe not wildly, but at least a little bit. So then we wanted to delve even a little bit deeper, and these are small sample sizes, but I was very curious. We were comparing people with cognitive impairment to those who are cognitively healthy. But what if we look more specifically at people with amnestic MCI versus being cognitively healthy, and those who have dysexecutive MCI versus being cognitively healthy? And what we see is that there are some variables that are unique to having amnestic MCI, words related to cognitive processes, and this measure called authentic that we get from LIWC. Negations, though, is common to all of these different subtypes. And so we can learn a little bit more about, we can think a little bit more about why that might be, but also it tells us that maybe this would be a good tool for distinguishing between these different subtypes and helping clinicians to determine what are the best therapies that they could recommend for someone. And so the initial analyses were focused on classification, but I'm really interested in how we can predict incident decline. How can we catch differences when someone's cognitively healthy? And so here we looked at whether these PFS scores predict who is going to decline over time on a cognitive screener. The one that we use in our studies is called the Telephone Interview for Cognitive Status, or TICS for short. And again, this was over a relatively short period of time, 4.5 years, but we see that the PFS delayed score was significantly predictive of who would go on to have cognitive decline. And the estimate here is a negative 0.05, but if you think about the wide range of values here, even a change of 100 in your PFS score, because in this score, it's really the tails that are the most meaningful. So the very upper end is the most meaningful. So if you think about a change in PFS of 100, Now you're talking about a difference of 5 points on this cognitive screener, which has a maximum score of 51. So it's actually pretty significant, and it's only over 4.5 years. So the conclusion from that analysis was that these subtle linguistic features are informative for detecting cognitive impairment and predicting decline. And again, what you see here, I think this one's reversed, but the black text here is what's traditionally scored, and the red is everything else that we might be able to read more into using other methods. But I was always wondering, part of the instructions that we read to the person when we give them the logical memory task is try to remember it just the way I say it, as close to the same words as you can remember. And I don't know the history of how that got in there, but I always wondered whether it was actually meaningful. And does worked mean the same thing as employed when people are saying this back? And so Sehal found this really great program called Stacy, which allows us to look at semantic similarity. And if you want more details on this, he's definitely the one to go to. But what it does is it tags each word in a sentence, and it gives a numerical rating to, it assigns it a value based on its word features. And then it gives you values of how the word is embedded in sentences using sentence vectors. And so 2 sentences that are closer on this vector scale are more similar. And so the values range from 0, not similar at all, to 1, completely identical. And we applied it to looking at similarity of paragraph recall. So if we read the person the logical memory paragraph, then immediately they tell us what they remember from the story that they just heard. And that's an example of someone's output. And then 30 minutes later, we ask them to recall it again. And again, they have output that usually has changed at least a little bit. So looking at similarity, of course, right off the bat, we're interested in how similar their immediate recall is to the stimulus. And this person's similarity score was a 0.95. And in the histogram, you can see the range of scores for all of the participants. And then, of course, we are interested in how their delayed recall is similar to the stimulus. This person got a score of 0.92, and you can see there's a little bit more variation now in the histogram. But then I've always been also interested in how someone's delayed recall changes from their immediate recall, especially because I work with centenarians and they have a lot of hearing impairment. So a lot of times their immediate recall is poor. And I'm not so interested in that being poor. I'm interested in whether they lose information over time from their immediate recall. And we call that retention. And this person's retention similarity was 0.97. And when we look at each of these similarity scores, the immediate, the delayed, and then this retention one, all of them are significant in predicting cognitive impairment when you put them into logistic regression models with age, sex, education. And then because we are a family-based study, we also have to adjust for relatedness of the family members. But then when you put all 3 of them in the same model, it's the delayed recall that really is the most significant, similar to what we were seeing with LIWC. And actually, we added MMSE, a cognitive screener, to the model, and it still was significant above and beyond MMSE, suggesting that there is room for this to be predictive of, or to help with us classify cognitive impairment even beyond a cognitive screener. And so again, we have precision recall curves. The delayed similarity in demographics is a 0.77. Once again, logical memory, the traditional score is actually doing a little bit better. There are some caveats to that. How we determined whether someone had cognitive impairment or dementia or normal cognition was based in part on their traditional scores. So it would be pretty surprising if our digital metrics could outperform that. But actually the best performing model, again, is the one that includes the similarity score in addition to the logical memory traditional score and a cognitive screener. So it's adding information that is clinically useful. And when we now switch to longitudinal analyses over about 4.5 years, looking at that same TICS performance, we see that the delayed similarity score does predict who is going to decline. And so we've really, SEHO has done a lot looking at lexical features. We started to look at acoustic features using the repetition task on MMSE, no ifs, ands, or buts. So it's about 2 seconds long. And we were using Open Smile with many, many variables. And it was very complicated. And we learned that there's a lot of specialized knowledge that's needed to interpret the results of the data. So I would say we are swimming in variables and data and variables. And we're looking for new collaborators to help us interpret the wealth of information that we can extract from these files. Yeah. So in conclusion, okay, good. Digital technologies, I really think, are so promising for helping us capture subtle, clinically meaningful cognitive behaviors. I'm extremely interested in the potential for them to identify preclinical things that predict who is going to go on and become impaired. I think they have the power to increase the specificity, not just saying that someone's cognitively impaired or not just saying that they don't do well on a test, but is that due to motor considerations? Is it due, maybe we can even extract some measures related to vision or hearing, or is it related to cognitive changes? I think where we're seeing similar performance in some of these off-the-shelf digital metrics, they're performing similarly to the traditional metrics. That suggests that we could really use these tools to automate scoring, and especially if you think about in the case of in a clinical setting where a primary care physician does not have specialized knowledge to score the time to score these tests and interpret them. If you could administer a 90-minute digit symbol substitution test and get a lot of important clinical variables, that would be a great setting to do it. Or if people want to administer cognitive tests to themselves online, some caveats to that. But I think this really opens the door for that. And then there are a lot of open access tools out there. We are trying to find out new tools that we can apply to our digital files to get more metrics. There are a lot of constraints with HIPAA compliance, especially in terms of the voice files. We can't just upload our data somewhere and use tools that are in the cloud. Unfortunately, it has to be a tool that we can download on our computers and do the analyses or do the feature extraction in a secure environment. And then, as I mentioned, a lot of these results take specialized knowledge that I don't have. I have a lot of neuropsych knowledge. I do not know anything about acoustics. And so we're always looking for more collaborators. And thank you very much.
Amazing. Let's start with questions here.
I have a question. You taught in one of my classes, so this is some of this stuff is just familiar to me. But you mentioned that you could grow neurons from these centenarians and then amyloid and see how these neurons would react. Have you done that? Or is it just like
Our collaborators are working more with the microglia right now, so kind of trying to understand how the inflammation is different in these superagers.
That's really cool. Yeah.
Yes, they have. And these induced pluripotent stem cells, we're talking about ends of, they're looking at 2 cognitive superagers versus 2 other centenarians. So it's very small sample sizes, and we're still, right now, it's very early phases to make any grand claims, but they do seem to act differently.
Can you follow up on that with that really amazing slide you showed where the middle-aged folks relative to older adults, the brain is so different versus the septuagenarian versus middle-aged folks? What is your theory of what is happening in these brains that have aged? I mean, there is aging going on. It's just that it does not show in cortical thickness because microglia is fixing old neurons as fast as possible. Like, what do you think is happening in those brains?
So, I mean, we believe that the inflammation processes are different in these superagers, and that also their amyloid and tau resistance, I'm sure, plays a big role in that. Their synapses are different. We're looking at synaptic density. So they seem to be operating differently on many different fronts. And I think a big question is whether maybe they started out with way more synapses than other people. We don't know yet. Or maybe they had thicker cortices to start with. I think people are leaning a little bit more toward they actually had the same as other people, but didn't show any change over time.
And so what about all the, I mean, that makes sense to me, but what about all the other age-related things that happen to these brains, like small vessel disease, small lacunae? They get all that, but they seem to overcome it or recover from it?
Yes. One of the really interesting things about the centenarians in particular, because we've gotten 15 brains so far, and all of them have cerebrovascular disease. Wow. So somehow they are managing to not feel the same effects of it. We do see some slight executive dysfunction among the centenarians, but some of them are retaining this exceptionally high episodic memory performance. And they have things, some other rare pathologies, things that get more common as you get older, RTAG, latency. Some of them do have amyloid and tau at intermediate levels that would cause dementia in younger people, and it's not causing it in them.
I have a question. That was a fantastic talk. Thank you very much. I think it was the first study that you mentioned. It said majority white and maybe like 60% or 80% females. Yeah. So I was wondering if someone has looked into that a little bit more, if there are any like ethnic differences, sex differences. And then I had a follow-up question about genetics. So I'm very interested in genetics and PPA. So for instance, in MAPT, there is a lot of like literature on certain like regions in Europe with like really strong family histories. So I don't know if you have any thoughts or results.
Yeah, so women live longer than men. So actually 85% of our centenarians are female. The interesting thing is that although the women far outnumber the men, the male centenarians are more robust than the women. They are the ones who are maintaining perfect cognitive function and physical function. So they're rarer, but they tend to be a little bit more robust. The females are a little bit more able to live with cognitive impairment. They're a bit more resilient, I would say. All of our samples are about 96% non-Hispanic white. And I would say that's more about recruitment bias and the way that we go about finding centenarians and these long-lived families. So we use voter registries and large mailings. For the Long Life Family Study, we used mailings to people in the Centers for Medicare and Medicaid Services. So it requires people being proactive and responding to our mailings. And so now we're doing a lot more community outreach, attending community health fairs, talking to people about our study. It's really great to get the word out about our study anyway, but we're also trying to find centenarians who we might not have captured using those standard recruitment methods. And then the genetics. With our centenarians, we haven't found 1 or 2 genes. It's really more like 200 genes with small effects. And it's like winning the lottery. Like you have to have all the numbers in a row or all these different genetic variants, and that's what helps get you to these really extreme ages.
Vinod has a question for you, sorry. Do you want to read it or do you want to unmute yourself and ask your question? Or?
Sure. Hi, Stacy. Hello, everyone. So, Stacy, I'm just curious. As you probably know, I also have had a bit of the Edith Kaplan background, and I when she was alive, I had this chat with her because my interest in informatics and neuropsychology and hers in the process approach, her interpretation of it was always from a psychometric standpoint. So my question is, in what you term thinking time in the patient's the clock drawing, can the digital pen pass out the numerous possibilities of spatial operations underlying these drawings, or the spatial motor transformations? Because from a purely cognitive point of view, that is a very very compelling question. We could have the same kinds of drawings, and underlying these drawings could be very, very different spatial processing going on. So I'm just curious if there's any indication that the digital pen can do that.
In the clock drawing test, there's a company now called Digital Clock, DCT Clock, and they have all different metrics.
You mean Linus?
Yeah. And they have all different metrics, and one of them they're calling spatial aspects of the clock. I think it remains to be seen how valid that measure is. I wouldn't say that you could parse it out from thinking time per se. That seems difficult.
Okay. If I can tack on another completely unrelated question, this is to do with your final slide and the HIPAA issue. I'm facing the same kinds of questions right now with the tool being developed with CDS and Spark at BU for spatial encoding. So maybe we can probably take this discussion online. But if you de-identify your data, is it something you still cannot transmit over the internet?
So we can easily de-identify the pen files. We can erase where the ID is written on, but the voice files, I'm not convinced there's a great way to do that yet, because I know that you can like mix up the voice, but it also can be unmixed. And so I've been hesitant to share those data.
Okay, great. Thank you. Thanks.
The example I'm told all the time is you can always recognize Morgan Freeman's voice no matter how you hear it and where you hear it. So
That's a great one.
it's just that's why that's the whole thing about PHI. But your question about thinking time, like I feel like that's where the goal is. Like if you could isolate thinking time and show that that's actually a diagnostic biomarker in some sense, that people eventually get to the right answer, but they just take so long to get to it, and you capture that in the pen not being moving time. That's what you're trying to get at, right? Like you can isolate that.
The thinking time, yes. I don't know about spatial characteristics and even visual scanning and things like that. I would love to be able to integrate eye tracking software. There's so many things we could do with digital data, and that would help us further parse out when are they thinking on that DSST test, when are they consulting the key at the top, how often are they consulting it?
Yeah. I mean, we're past 5, but if you have your question, you should ask it.
I can wait until afterwards if you'd like. Or I can ask it now. It was related to thinking time.
Go for it.
Okay. Well, this was incredibly fascinating, by the way, and I think you have such a rich data set, and I'm very interested in the whole concept of being able to not just look at these centenarians, but also their families, and not just from a genetic standpoint, but what habits might be being passed down. And at the same time, I was fixating on this clock draw task, because if you start looking at these younger generations, is their thinking time going to be longer because they're so used to these digital versus analog? And similarly with writing time, because there's more texting and typing, and how you would maybe tease that out from other variables that you might be trying to look at here.
Yeah, I always get this comment about the clock drawing test, and it's certainly valid. At the moment, it seems like it's still working in the younger generation, but probably I wouldn't be surprised if they had longer processing times of how do you set the hands to 10 after 11. But that's why I would never use only just one test to extract my digital metrics. Just like we never want to administer just one test in an ideal situation, we want a whole complex of tests that tell us different things. And then we can see across performance. Because even on a digit symbol test, you can think about memory components to that, even though it's not a memory test. And so my hope with these digital metrics is that we can start to take out more features like this from every test and get a whole profile of someone's cognitive functioning, not just a score of 50 on this one test.
And further, if you start seeing those changes on something like the clock draw, it might correspond to changes on some of these other assessments too, just based on maybe there are fewer working memory demands just in day-to-day life nowadays than there were. And it would just be cool for you to be able to sort of pinpoint when that change is happening because you're tracking this longitudinally. It's cool.
And then I think more to your point also about a test not being valid as time goes on, something like digit symbol coding or writing even a sentence, people are less likely to be using a pen. It's more about how someone changes over time. So what does their sentence look like at the beginning and how does it change over time? What does their voice sound like at the beginning and how does it change over time?
Yeah. Very interesting.
And that's why studies with longitudinal follow-up, that's where the gold is.
Okay, well, it's definitely past 5, so we should thank you again.
Thank you.
Thanks for